share with you a topic that grabbed my attention this last week. It's about the ultrasound activation of drugs. Imagine a world where a medication could be activated in a targeted area of the body by ultrasound equipment. This ultrasound equipment amplifies signal to a certain spot in space. I think this lowers the potential for side effects because it keeps the activated form in certain regions, and that active form is more likely to wreak havoc in other areas of the body. The study that I'm going to share with you is from labs in China, Germany, and the Netherlands. It was published in Nature Chemistry. This study may actually be the start of a new field called sonopharmacology. The particular sound that is used to activate the molecules is ultrasound. And ultrasound is defined as signal that's just out of the reach of the human perception and on the higher frequency, 20 kilohertz, give or take. For me, I like to think I have excellent hearing, so maybe 25. Ultrasound can actually break covalent bonds and non-covalent bonds. It also can disrupt interactions because of microbubbles that form due to ultrasound. In this study, they actually used 20 kilohertz frequency, and the power they use, the intensity they use, ranges from 15 to 36 watts per centimeter squared, which means energy in joules per second per centimeter squared. Our microwaves are about 800 watts, so this is much less than that, considering that the probe that they use is 1.3 centimeters. They did pulse sonication, 1 second on, 1 second off, or 2 seconds on, 1 second off. Okay, so their first demonstration of ultrasound activation was shown with an anti-cancer drug called camptothecin. In this case, there's 2 sulfur atoms that get cleaved by the ultrasound. After cleavage, a 5-exotrig cyclization happens. I just thought that was a cool name. 5-exotrig. Sounds pretty cool, right? So this R group that's shown can either be the anti-cancer drug, or it could be a dye that is used to monitor this reaction. One really cool thing about this result is that this anti-cancer drug is actually pretty insoluble. So the polymer that is used can deliver the drug more easily to a tissue where it can be activated. Lots of great things about ultrasound activation. They showed that the activation with the dye over 4 hours gave 80% conversion using absorbance and fluorescence as a readout. They showed that they were able to activate the anti-cancer compound, camptothecin, by sonication before adding it to HeLa cells. When they added the sonicated inactivated compound to HeLa cells, they saw a response and a decrease in viability. The next example of ultrasound activation was shown with aptamers. Aptamers are RNA molecules that fold to form a certain shape or structure. Aptamers will bind small molecules. In this case, they bind neomycin and paromomycin. These are actually aminoglycosides I studied in grad school. I studied one step in the reaction or 2 in the biosynthesis of neomycin. The researchers in this study used the R23 aptamer. They used rolling circle transcription to form the polyaptamer, so it was repetitions of the aptamer over and over in one long strand of RNA. Ultrasound is able to disrupt the phosphate backbone and also the aptamer structure. With the agar diffusion test, neomycin was shown to inhibit growth, while tests with the polyaptamer with neomycin did not. In this test, they were actually able to activate the aminoglycoside by exposing the plate, which is growing Streptococcus aureus, or the pathogen, to ultrasound. They were able to expose the bacteria to ultrasound without killing them. This was not true for the HeLa cells. They found that 1 minute of ultrasound released 40% of the aminoglycoside, and 30 minutes released 80% of the aminoglycoside. The third example of ultrasound activation of small molecules for therapy is the gold nanoparticle with the data peptide and vancomycin. Vancomycin is not a polymer, it's a small molecule, but it's a large small molecule. And it binds to a short peptide, which is actually smaller. In the study, they tethered the peptide to a gold nanoparticle. Gold nanoparticles were chosen because they're easy to functionalize. You have the ability to choose a size, and apparently it has a good ability to bind sulfidyls. I've never worked with gold nanoparticles. The data peptide has a sulfidyl on one end, which is bound to the gold nanoparticle. The gold nanoparticle is protected by citrate. After sonication, only the nanoparticles with PEG on them was shown to inhibit bacterial growth of Streptococcus aureus. Without sonication, there was low inhibition. Without PEG, there was inhibition. These researchers were able to get more sensitivity to vancomycin in a cell-based assay by conjugating vancomycin to the gold nanoparticle. In this diagram, each gold nanoparticle is actually covered with biomolecules. The large gold nanoparticle is covered with data peptides, and the small gold particle is covered with vancomycin. So you can imagine that huge aggregates get built up as the particles with data on them are drawn to the nanoparticles with vancomycin conjugated to them. They were able to see these aggregates with transmission electron microscopy. They were able to break up these aggregates by sonication, by disrupting the interaction, the non-covalent interaction between data and vancomycin. And when these aggregates were released, they inhibited the growth of Streptococcus aureus. Interestingly, when we build up these aggregates of nanoparticles, the absorbance of light changes due to Mie scattering. Mie scattering happens when aggregates get near or just less than the size of the visible wavelength of light. And so the absorbance of the vancomycin was increased. It was redshifted by about 100 nanometers. The paper claims that in order for ultrasound to be used as a targeted activation for therapies, we need to be able to control the time, the frequency of the ultrasound, and the energy to minimize damage to tissues. Ultrasound is actually already being for prostate cancer in a method called HIFU, high intensity focused ultrasound. This may make the application of drug activation easier for development. But the mechanism for prostate cancer is totally different. HIFU uses ultrasound to kill cancer cells, whereas this will activate a drug to treat the tissue. I don't think that we know yet if we can develop a drug easily that will require low enough energy for effective activation of a drug. It seems plausible based on this research, although the timescale is on the order of hours, which is not exactly convenient for a patient. Also, can the method be specific enough to not disrupt essential function in the targeted tissue? That wraps up this video on ultrasound activation. I'm just starting out this video series. Let me know what you think in the comments. Take care.